Okay, let's begin. Thank you all for coming again. We've talked so far about what evolution is. Remember what it is? Give me change in what? Yes, change in allele frequencies over time. We've talked about how it can occur through genetic drift, migration, mutation, and natural selection, where natural selection's the most important one, the most interesting. We know what that is, right? Three things. Has to be heritable. That's number two. There's got to be variation for a trait. And the most important of the three, differential reproductive success. So we've talked about all those, but we haven't actually talked about the genetics yet. And if you're talking about a change in allele frequency, you have to be talking about what are alleles. And that's what we'll do today. We'll talk about what they are physically and how they behave physically so that we end up with this property of alleles, the way they get passed on from one generation to the next, that we see. In order to do this, we have to talk about Mendel. And Mendel's a tough one to talk about because everybody has heard a little bit about Mendel at some point in their life. Almost everyone has a picture or an idea in their head of Mendel and what his work was. Almost always it's incorrect, though. What do we know about Mendel? So I want to offer up. What do you know about Mendel?
He and Darwin put their ideas together.
He and Darwin put their ideas together? No. Close. That was close. If they had met, they would have. And because their ideas wanted to be together, but they weren't. Tell me, they should have, yes. What else? Tell me something about him that's true. He did genetic stuff. You could even say he invented genetics, so that's a good answer. What else do we know about him? He worked with peas, yes.
At Abbey?
Yeah, some monastery somewhere. Yeah, so we actually figured out that whole, some of them are recessive, some are dominant, so you can carry something but not have it show up in your phenotype. What else? He did thousands of pea experiments, yeah, so, which is part of the misconception. People imagine that he did a pea experiment. He did a whole bunch. I would call him the father of genetics, yes, although he was a monk, so there's probably then no mother of genetics. I'm not sure. But yeah, that would be okay. Here's the thing. Mendel, he was born a little bit after Darwin, so early 1800s, like 1822 or so. And he went to a monastery. We tend to have the idea, or maybe it was just me, but we have the idea because he lived in a monastery. I imagine that he's by himself, he's in total isolation, and that when we talk about him doing pea experiments, I imagine he's got, 4 little pots on his windowsill, and he's doing those. And it's not exactly how it was. He went to a university in Vienna. This is his monastery was in a place called Moravia, which is now where the Czech Republic is. But it wasn't in total isolation. So he went to university. He was trained in biology. And he then decided that he wanted to join this monastery. So he went there. The head guy, I've got a picture of all the guys from the monastery. So this is Mendel in the back row, standing up. Second one in, he's holding his pea plant. And so the head guy, the guy with a big cross around his neck, Abbot Knapp, gave Mendel an assignment when he got there. You would have an assignment. It would be, okay, I want you to do this, or I want you to do this. He assigned Mendel to study heredity. He said, this is an interesting scientific problem, and I'd like you to work on it. Now, he got going then on these plots, and he did experiments that involved thousands of individual pea plants. He had multiple research assistants working for him, so other monks who were there who would help him out. So it was much more like an actual research lab that you would do a 199 in, and then just 4 little plants on his windowsill. They had, what else do we know about their life there? Apparently they had a great wine cellar at this monastery. Mendel smoked 20 cigars a day. And he tried to be a biology teacher, so there was a nearby town they could earn money, and so he wanted to be a biology teacher there, and he took the exam that you have to take. You have to get certified, failed the biology exam twice, and so for his entire career he taught physics at the local high school because physics is easier than biology, apparently. But let's talk about the experiments that he did, kind of, and where they fit in. So Darwin wrote his most famous book in 1859, The Origin of Species, and in this book, as we know, he said that a trait can change over time within a species, actually within a population. We shouldn't really use those interchangeably, but within some relatively isolated group of organisms, if you've got variation for the trait, it's heritable, and you have differential reproductive success. So we already know all this, but it's important enough to write again. Darwin had huge amounts of data on variation for a trait. How many of you went and looked for Darwin's book online? I told you to. Oh, come on. Just go online, look for it. Remember I told you, there are 4 chapters is all you have to read, and you will be considered very literate by your friends. Read just bits and pieces of the first 4 chapters, skip ahead to the very end, read the last paragraph. Last paragraph is beautiful. Someone over here did actually read it. Did anyone look at the last paragraph? Did you look at it?
Yeah, I checked it out.
Was it beautiful, the last paragraph?
Yeah, well, it shows how much he really appreciated the life that he was studying and stuff.
Yeah, he really realized that even though everything he was learning was coming in conflict with what he had known before and it was going to rock the world, he said, this isn't all bad. This is okay. His famous quote is, because people were worried that as soon as you took the view that evolution shaped the way species looked that were on the planet, that was terrible because all of the idea of beauty that had been created in the world, et cetera, would go away. And he says, no, there's grandeur in this view of life. He thinks that just by understanding how organisms get changed, that's cool. It replaces the previous view, but it's nice. So just take a look for it. Come on. All right, so he knew that there was variation for a trait. So as I said, the first 2 chapters of the book, he goes on and on, just looking around saying, look, all the individuals vary. Sort of like I would look at a classroom here, and I can see you all look different. That's something that was important to him. Heritability. He knew that offspring resembled their parents. Didn't really know how, so he just kind of left it. But then he said, if out of all that variation that we see, some of the individuals get to reproduce more, others reproduce less, the alleles, the genes that code for the traits that cause them to reproduce less, they kind of fall by the wayside. Their market share in the population is reduced. Darwin couldn't figure out heritability. He knew somehow there was a way that offspring resembled their parents. He knew that something caused that, but he didn't know what it was. In his book, he just said, hey, we know this is true. I wish I could figure it out, but I can't. Let's just leave it as number 2, and we'll say, assuming it's true, blah, blah, blah. So this is where Mendel came in. He did all these experiments. We'll talk a little bit about them. And he was aware of Darwin. Everyone in Europe was aware of Darwin. And so Mendel realized, he's like, I'm going to figure this out. I'm going to figure out how it is that offspring, whether they are offspring of pea plants or offspring of humans or pigeons or anything like that, I'm going to figure out how it is that they come to resemble their parents. So he did a bunch of experiments over the course of several years, collecting, as I said, thousands of data points. And over the course of these experiments, very clever experiments, he figured it out. He figured out, aha, here is a way that if things work this way, it explains why offspring look like their parents. And he published the results of his talk in a journal called the Journal of the Natural History Society of Brünn in 1865. So this is right about 6 years after Darwin's thing has come out. This was not an obscure journal. This was all the major libraries in Europe had it. People read these journals. Not only that, his paper was cited by lots of people. When you write a scientific paper, as you go through and you make various assertions, you put references to who said it before. People cited Mendel's paper. So it wasn't like he was just off in obscurity somewhere. People were aware of it. Anyway, so he then was invited to give a talk at this Natural History Society of Brünn. And this was in February of 1865 over the course of 2 meetings. And I try to imagine what it was like, because at the time everyone was talking about natural selection. Everyone was talking about 3 necessary and sufficient conditions. Nobody quite understood what heritability was. And he was going to talk about it. So I gave a talk once at this group called the Cambridge Entomological Society. And I'm not particularly interested in bugs, but they invited me to talk about some research I was doing. And the thing about this group was that they'd been around for more than 100 years, and they had these giant log books that you would open up, and every time they would meet, it was a bunch of retired old guys who would sit around the table and they'd bring in, I found this bug in my backyard, I found this new honeybee, and they'd pass it around and they'd talk about it. But in their giant book, they would record, Jay Phelan came to speak on I forget what it was, maternal age effects in milkweed bugs. So I gave a talk, and everyone writes down who's there, and then they write about the talk. Anyway, Mendel went to this society, gave a talk. They have the same kind of book. And he gave the talk, and this is the saddest thing I can tell you. So they still have the logbook, and in the logbook it says this for February of 1865. Gregor Mendel spoke on inheritance in pea plants. There were no questions. That was it. He gave this talk where he said, here's what the genes are, and here's how the alleles get split up, here's how they get reestablished. And he answered Darwin's question of how does heritability work. And remember, this was a bestseller, Darwin's book, so this should have been people going nuts, talking. The room should have been full. Everyone should have been excited about it. Nobody understood it. They just weren't ready for it. And so he did experiments for a couple more years after he did this talk and published the paper describing these. You can actually read his paper on this as well. It's amazing. It reads like an introductory biology textbook. It's really clear and describes it. But people just somehow were not ready to understand this yet. So after about 2 more years of doing ever increasingly elaborate experiments, Abbott, Knapp, and the others at the monastery said, hey, you might want to take it easy on these experiments. You're using up a lot of resources here. And, there were no questions at your talk. Just ease up. And so Mendel got progressively depressed and lost confidence that he had done something important. And he took a position higher up in the administration at the monastery. He stopped doing his experiments. And over the remainder of his life, he became less and less convinced that he had done anything. And then he died. That's it. Isn't that depressing to have done something truly amazing? It's the father of genetics, we're saying 150 years later, and he died thinking he was a failure. Why don't you just go? I get really depressed for him because his paper is very good. Let's see what he did. Fortunately, today we're ready to understand it, I think. Certainly more so than they were at that time. So let me draw out this. Okay, what is that? California? No. A sock? No. Pea pod? Get on? No, it's a chromosome. Mendel actually didn't. That's 2 now. Sorry. We'll call this chromosome number 14. In all of the cells of your body, more or less, there are a couple of exceptions, but more or less in all the cells of your body, you've got 2 copies of every chromosome. So you have a genome, which just refers to all of your genetic material. In humans, the genome consists of 46 chromosomes, but they're divided into 23 pairs. So you have 2 copies of each of these pairs. They're called homologous pairs, homologous, homologous. And the homologous pairs have all the same genes on them, And we refer to them as, the maternal and the paternal. So for chromosome number 14, you have inherited from your mother one copy and from your father a copy as well. Now this chromosome is going to have, it's a long sequence of chemicals. It's DNA. We're not going to concern ourselves too much with the structure, but it's like a code. It carries information the way your computer hard drive has zeros and ones, but the order in which they're held together conveys information. Well, the sequence of chemicals in DNA also carries information, and at a particular location, you've got a bit of information that tells your body how to produce some product, and we would say that the locus equals location of a gene. So we would say at some particular locus. So here's just a bunch of the chromosomes. So I said in humans there are 46 chromosomes divided into 23 pairs. We number them 1 through 22, and then the last pairs are called either an X or a Y. You can have 2 copies of the X, or you can have an X and a Y. And so if I've drawn number 14, actually they have more of a smudge there. They're not actually in color. This is fake color. But you would say you've got the location of a gene would be the locus. So I'd say at this locus, an inch down from the top of chromosome number 14, you have a gene. And that gene is going to be the instructions for how to produce something. So you might say, get a different color. Gene for milk digestion. So this sequence of chemicals right there carries the information for an enzyme that if you have lactose in your body, it can break it down into 2 simple sugars and extract energy from it. We might also say, okay, there's a gene for hair color pigment, or there's a gene for a chemical that causes your bones to get bigger so that it's related to your height or size or something like that. Now, here's the important thing, is this. You can have, in your mother, she might have given you the allele for normal digestion. In other words, it tells the body, here's how you produce lactase, and everything will be fine. Your father may have given you a slightly different one, a mutant allele, no digestion. So it produces a chemical, it's a set of instructions, but there's something wrong with it a little bit. So the chemical it produces can't break down lactose. If you consume lactose and you don't break it down, you're going to get sick. This is an important thing, that the same gene is on each of the homologous chromosomes, but because you can have a different allele for it, they're not exactly the same. That's why I can say there is a gene for eye color. That tells you how to produce the pigment, but the allele might be blue pigment or brown pigment. Question in the back. Say that again? What kind of what? Milk? Good question. I just lumped together human milk, probably cow's milk. In this case, I'm thinking of there is a lot of variation for ability to break down cow's milk. Probably goat's milk as well, sheep milk. It's a pretty generalized all-purpose enzyme. If you have it and you have lactose, you can break it down. Question?
Does each chromosome have a different section of
Okay, so that's a good question. Does each chromosome have a different section of their DNA? Yeah, so you could think of your DNA as starting here on chromosome number 1, then going here on chromosome number 2, then there on chromosome 3, there on 4, and you could sort of line them all up, and those are all of your traits. So the fact that you've got then a second copy of chromosome number 1, you just have 2 copies of each, but from 1 through 23, all of your traits are divided. Every chromosome has a different collection of traits, and the homologous pair has 2 copies of each trait. Is that clear? It's not the most obvious thing, right? Otherwise, everyone, when Mendel didn't describe exactly this, but people would have understood it, and they didn't. Anyway, so you've got this sequence of DNA that codes for milk digestion. Down here it might be for what blood type. Here it might be something about the texture of your hair. Here it might be something about your ability to break down alcohol. Question? What's that? Yeah, it's a good question. Why are they in the order they are? Doesn't seem to be any rhyme or reason to where they are. And over the course of evolution in different species, you'll have chromosomes. They split up, and some species have lots more chromosomes than others. There doesn't seem to be any rhyme or reason to it. What's important for you to always remember is that the homologous pair has the same gene at every locus, but each of those genes could be different because they could be different alleles. Now, you could have inherited the same allele from your father, in which case this sequence and that sequence would be identical, but not necessarily. You'll notice something here. This is really embarrassing even to tell you this. I was all the way in graduate school, and I was pondering, sort of like your question about are they just randomly assorted. I was curious how they named the various chromosomes, and it didn't dawn on me literally until graduate school that they just number them according to size. The biggest one is number 1, and as they get smaller, they're down. So number 21, 22, very, very small. So in different species, that's how they end up getting their number. Okay, so let's look at then a generic cell. This would not be a human cell because in this cell, each of those strands represents a chromosome. But I've drawn a purple one that's kind of has a little kink in it. I've drawn a green one that's curved. I've drawn a red one that's squiggly. So the 2 red ones that are squiggly represent a homologous pair. The 2 purple ones represent a homologous pair. How would this generic cell differ if it were a human generic cell? There would be 46 lines of some sort, and I would have had to use 23 different colors. Yes? Okay. In that previous one, I said one was the mutant enzyme for milk digestion, the other was the normal one. For most traits, that's the question about you have dominant and recessive traits. A recessive trait is one that if you have a copy of the dominant trait, the dominant shows up in the phenotype. So dominant traits mask the effect. For most biochemical traits, the 2 genes both get coded for. So your body produces the messed up enzyme and the normal enzyme. And so they're floating around your body. So in the case of that picture I drew where there was 1 good copy and 1 bad copy, that person would have half as much lactase as someone else. And that means if they consumed small amounts of milk, they could handle it. If they consumed huge amounts of milk, then they wouldn't be able to do it, to break it all down, and they would end up getting sick. With lots of the genes in our body, that's how they work. So there isn't really a good dominant or recessive. Both of them get produced, and if you have 2 copies, you have more of it, and you can do whatever that is with more. In a lot of cases, having at least 1 copy, that's enough. So in the case of having 1 good copy and 1 bad copy of the lactose-digesting allele, most people wouldn't notice the difference. They'd be like, yeah, I'm fine. If you had 2 bad copies, then you would notice it. So in this cell, how many chromosomes are there? Every linear strand of DNA that is discrete on its own counts as a chromosome. So you'd say that there's 6. How many homologous pairs? In that case, there are 3. Not all species have 2 copies of every chromosome. Almost every species we talk about this term will, so that's why we spend time on it. That condition is called being diploid, means 2 sets. Haploid means you only have 1 set of chromosomes. You could have 3 sets of chromosomes. There are no species that I know of that get by permanently with 3 sets. That would be called triploid. There are tetraploid species, 4 sets. There are a lot of those. You could be octoploid with 8 sets of everything, which also works, but your cells start to get a little bit cramped. All right, let's talk about what Mendel figured out. Mendel's primary results are broken down into 2 laws. He didn't call them Mendel's laws, but we do. The second one is not super important for our purposes. The first one is very important, but it's also so easy as to be almost offensive. He said this: a gamete is a sperm or egg cell. Gamete receives only 1 allele for each gene, even though the organism generally has 2. So you normally have 2 copies of every allele because you're diploid. You have your maternal and your paternal copies of the chromosome. However, during the production of a sperm or an egg, only one of those gets put in. In the case, let's say we have the genotype of the milk digestion thing that I wrote up there, where you've got one version that is normal functioning, one version that is non-functioning. That individual, if it was a male, half of his sperm that he produces would have the good version and half would have the non-functioning version. So then when he is going to fertilize an egg, whether or not he passes on the good version or the mutant version, totally random. But then at fertilization, because there is a female somewhere who is also diploid but produces a bunch of eggs that only have 1 copy of every chromosome and therefore 1 copy of every gene, when that sperm and egg come together, now you become diploid again. So I've just drawn a smaller version of our generic cell, 2 of the purple, the green homologous pair, the red homologous pair. When they produce a sperm, I've got a little tail on it there, or an egg, they are haploid. I should write that in. And when the sperm comes together with another haploid gamete, the egg, they fuse together, and now you get 2 copies of each. Question?
How does Mendel know about chromosomes?
Mendel actually didn't know about chromosomes. They hadn't invented good microscopes yet. And so all of this he was just figuring out from these experiments where he looked at which traits they had and he crossed them. So it was really cool. He was, it was as if he were told to solve a really complicated math problem, and then they said, oh yeah, you can't have a calculator and you can't use calculus or something like that. So he just figured this all out. Later on, it was verified when looking at them. So Mendel imagined, rather than there being chromosomes with a bunch of genes on them, he thought every gene was just free-floating. So you had the section, and you had 2 copies of it, and it was floating around. And then when you made a sperm cell, 1 copy of it went here, and 1 copy went to the other one. So it was essentially the same. He just didn't imagine that they were linked together. So this is it. This is Mendel's first law, and as I say, the one that I think is more important. And it's really simple, but what is powerful about it is that it now reveals, aha, that's how I can have some traits from my father and some from my mother, because they've each contributed 1 of the 2 copies that I have. Not only that, it tells me how I can have some traits. When I look at my father, he has a mother and father. And I can look at some of the traits that I have came from his father, some came from his mother, because when he was producing sperm, remember, he's diploid, 1 from his mother and father, and every sperm he produces has a gene that came from either his father or his mother. And so you see how at each generation, you merge two individuals' traits, but then when you produce your gametes, you are giving each of them only one of them. Let's say, hold on one second, you'll see pictures all the time of chromosomes, and they always show chromosomes looking like X's, and this is really frustrating to me because most people who haven't taken a lot of biology think that a chromosome looks like an X, and they don't look like an X. I've already told you, they look like a sock, or they look like California. Whatever it is a linear thing. It has a top, a middle, a bottom. It's not an X. The reason we see so many pictures of X's is that most of the time in your body, chromosomes, because it's just this tiny DNA molecule that's really, really long. Think of it like a really long shoelace. Usually it's all unwound in the cell the way a floppy shoelace would be. Right before the cell is getting ready to divide, it curls up. So like you take your long shoelace and you twist it and you twist it and you twist it and you twist it, and it gets shorter and shorter and shorter and thicker and thicker and thicker because the cell is going to divide. Normally when it's unwound, if you took a picture of the cell, you can't see it at all. It's too thin. So now when it gets thick, it gets easier and easier to see. But the problem is the only reason it gets thick is that the cell's about to divide. And before cells divide, every chromosome makes a duplicate of itself. So this linear thing first makes a copy of itself, and then it starts coiling in. So we look at this, and they look like X's, but that's actually a chromosome and an exact duplicated version pinched together at the middle. So that's a long aside, but it's just a way of saying you'll see it looking like an X. But if I say the locus is, X number of inches down from the top, on something like this, you'd be looking at, that would be the top, and that would be the entire thing. And right here, this is just an identical duplicate version. Those are going to get separated out later. Same thing here. Any of the pictures are going to look like that. So in the process then of producing sperm, this is a cross-section of part of the testes where the sperm are now maturing. They're growing their tail. They're getting ready to swim. Or here, I love this picture. This is an egg moving down the oviduct. And so it just kind of tumbles down. It's going to meet up with some sperm and get fertilized possibly. I have no idea how they got that picture. I lose sleep thinking about it. Let's say, what else we have here? Here you can get a sense that every one of those sperm is haploid. It has one copy of every gene, one copy of every chromosome. Same thing with the egg. Only one of them is going to get in, so that the ultimate resulting offspring is going to have exactly one copy from each. Here's the moment of fertilization, the egg burrowing in. Again, I have no idea how they got that picture. Okay, so Mendel did his experiments. Actually, though, we'll talk about a different one. Let's talk about a trait, albinism. It's very rare to see an albino in nature. Why do you think that is? Do you want to be born like that giraffe? In nature, it's very tough because it's a real flag to predators. And so when the alleles come together, two of the recessive alleles that are both copies of how to make pigment for your body and they don't function properly, you don't have the ability at all, and you look like that, and predation rates are very high. Let's say, though, that you've got two individuals here where you have an albino. So albinism is one gene codes for it. We'll call it M for melanin. That's the chemical that gives pigment. If you have two copies of the recessive version, so just one of the versions, then you're going to be albino. If you have two copies of the normal version, you're going to be normal. If you have only one copy of the uppercase one, you're also going to be normal. These would be called the genotypes. Remember, we distinguish between genotypes and phenotypes. Genotype is the underlying genes that are there. Phenotype is the physical appearance. So these would be the gametes. Because of Mendel's law of segregation, you carry two copies in the body, but you only produce one copy in each of your gametes. So the tail means it's a sperm, so we're saying the male was the albino. I put this down here just to remind you these are the female gametes, so those are eggs. Now, when these parents come together, the albino and the normal, what's the genotype of the offspring going to be? It has to be big M, little n, M, because all the sperm carry a little m. even though Mendel's law of segregation says half get one of them and half get the other, half get little m and half get little m. Or with the mother, half get big M, half get big M. So when they come together, write down the genotype of the offspring in your notebook. So it would be one big circle because this individual is going to have two alleles. Come on, write it down. So it's a big M and a little m, but you should put a circle around it because, just to remind you that it's all in one individual. So the genotype is going to be big M, little m. You can write them in any order. It doesn't matter. The phenotype is going to be normal because, like that lactose digesting allele, if you have one melanin-producing pigment gene, it has enough melanin production, it codes for enough melanin production that you look normally pigmented. So this normal doesn't mean that the exact same thing's going on as this individual where both genes are pumping out melanin. In this case, only one of them is. So now the question is, we've got these gametes, this individual does. What if we cross two of they're called F1, so we call P1 for the parents' phenotypes. F1 are the offspring. If we cross two F1s, show me what the cross looks like. When you cross them, you don't know what you're going to get. So you have to tell me any possible thing that could happen. So I'd write it this way. I'd say F1 cross is going to look like this. So we're crossing two individuals. They are both heterozygous. It's called homozygous if you have two copies that are the same. So you can be homozygous dominant, homozygous recessive, or heterozygous. So these individuals are both heterozygous. In order to do this, you probably remember this from high school, the Punnett square. Punnett square, named after R.C. Punnett, Reginald. It's very sad, his story, because I won't go into it. It's sadder, possibly sadder than Mendel's. Now I can't. All right. No, but Punnett lived in the early 1900s. And he was a really smart geneticist, and he published a bunch of really interesting papers showing how different genes interacted with each other. And he had a bunch of students of his, people that he trained. And of his students, multiple students of his won the Nobel Prize. And he was as smart as they were, and he did as important work almost as they did. And he gets remembered for this thing that is like third-grade biology, the Punnett square, because it was his little way of keeping track of things. I mean, I guess it's better than nothing, right? We don't talk about the Alfred Sturtevant square. So you'd say this. You'd say you put the female gametes always go on top, male gametes go on the left side, and you say, all right, the male has a little m or a big M. We don't know which of those sperm is going to be involved in this fertilization. Same thing for the female. She's got a big M and a little m. We don't know which one. If that gamete and that gamete came together, here's what the offspring would look like. The genotype would be heterozygous. Phenotype would be normal pigment. If this sperm fertilizes this egg, it would look like that. If this little m fertilizes this one over here, it would be little m, little m. In that case, it would be albino. If this big M fertilizes that little m, big M, little m, and the phenotype is going to, again, be normal pigmentation. So that's how you figure out if you cross those offspring. You'd want to know, in the case of most crosses, what are going to be the ratios of genotypes that we see. Here's a genotype, MM, big M, little m. Here's a genotype, big M, little m. So those are the same. Here's a genotype, big M. That's different. Here's a little m, little m. So you would say 1, big M, to 2, big M, little m, to 1, little m, little m. Or 1/4, 1/4, 1/2. You do it anyway. What are the phenotype ratios here? 75-25. Yeah, exactly. You're good. For every 4 offspring, it's going to be 3 normal, 1 albino. And so that's enough for us to be able to say this first take-home message that Mendel's law of segregation points out that the gamete gets only one of the alleles from the pair that the organism possesses. Sorry, this is called mid-digital hair. You can look down and see if the person next to you has it. There are lots of traits that don't behave with complete dominance, where you either have the dominant or you have the recessive trait. There are, if you take LS4, you go on and on about incomplete dominance or codominance. And I'm not going to spend any time on those because they're actually, I think, not that useful. I think the way I described it, that you have 2 copies, and they're both cranking out production of whatever the product is, but usually having just 1 copy is enough to show the trait. But if there is ever a case where, let's say it's a pigment-producing thing, so sort of like the melanin, but it's in a flower where it's producing a reddish pigment, and the other gene, the mutant gene, is white pigment. If that's the case, if you're heterozygous, you might actually look pink, because you don't produce enough, whereas if you have 2 reds, you're fully red. So in that case, people tend to call that incomplete dominance. But our model that I described, where they're both cranking out their product, and you just see whatever the end result is of the mixing, that's generally how it works. So with...
I just remembered, like, the example for, like, a calico cat, I guess, was, like, a mix of, like, 2 normal cats, and then they, like...
Yeah, the genetics of calico cats is really complicated. It's not just about 2 genes having some interaction. There are lots of variations on these. But looking at it this way, that every gene produces some product and the interaction of them produces some net effect, should work for us. So I talked about lactose digestion. People would say, oh, the normal gene is dominant, right? As long as you have 1 copy of it, you can drink milk. But the way I described it wasn't really true, right? I said you could drink some milk, but if you drank a real lot of milk, you'd get sick. Whereas if you have 2 copies, you wouldn't. So I'm defining dominance and recessive almost as something that's not quite true. Here's another question that I at least have all the time. I talk about traits like lactose digestion, about albinism. Are those realistic? Are they representative traits? In other words, are there lots of human traits that are coded for by just 1 gene? Or, like, my gut feeling would be, no, they're probably all controlled by lots and lots of genes. But the answer is no. Or the answer is yes, there are lots of human traits that are coded for by a single gene. So I've given you a couple of examples. But the more we look, the more we find individual genes that code for visible traits. I'll tell you about one. Maybe you've heard about this in the media. It's a very depressing example, but it's something called fish odor syndrome. So you pretty much already know what I'm going to talk about here. This is a real disease that individuals have. And so here's from an article that was in a bunch of newspapers not long ago, but you see this occasionally. 2 competing teams of scientists have identified genetic defects that cause fish odor syndrome, a distressing disease that makes people smell like rotting fish. People with the uncommon disorder can be ridiculed as children and grow up to be lonely and depressed. These people lose their jobs, they lose friends, they're often very isolated. said Dr. Eileen Tracy, who led one of the teams that found the flaws in a single gene. Robert L. Smith of Imperial College of Medicine in London, a member of the other group, told of a woman who divorced her husband because she thought he was not keeping himself clean. When she learned he had the disorder, she was full of remorse, he said. No cure is known for fish odor syndrome, blah, blah, blah. The British research team reports its work in the December issue of the journal Nature Genetics. The newly found defects sabotage production of effective forms of an enzyme called FMO3. The liver needs that enzyme to produce a smelly protein called TMA, or trimethylamine, which is produced by, to process a smelly protein called TMA, which is produced by bacteria in the gut. When TMA goes unprocessed, it seeps out in a person's breath and perspiration, causing the offensive body odor. Everybody has 2 copies of the FMO3 gene, one inherited from each parent. The disorder arises when both copies are defective. It appears shortly after birth. Mothers find an affected baby smells a bit odd and may bathe the child several times a day in the next few months, Smith said. The baby's bedroom may have a heavy, slightly objectionable smell, particularly around the curtains and furnishings. Later in school, the child is often ridiculed as stinky or fishy, said Smith, who said children have been forced to leave schools because of the derision. Anyway, it goes on and on. It's an awful condition. So here's the thing. Very sad, but it fits in exactly with our model. Somewhere on one of your chromosomes, you have a sequence of DNA that codes for the instructions for an enzyme that breaks down this protein that bacteria that live in your body produce. So the bacteria are going about their business, eating stuff inside you, and as one of their waste products, they produce this stinky thing. Well, you have 2 separate genes that produce the enzyme, FMO3. They break it down, and it goes away, and you don't smell. So if you carry 2 bad copies of it, then you're not able to break any of it down. It seeps out, and you smell. What happens if you're heterozygous for the FMO3 gene? You don't stink because that 1 copy of the gene produces enough of the enzyme that you're okay. So you might have the hypothesis, well, if somebody had 1 copy of it, if you were really, really sensitive about smell, you might detect a tiny, tiny bit. Maybe. I don't know. Because they tend to only produce half as much of the enzyme. Anyway, there are other genes. This is called mid-digital hair. It's a bunch of hair growing below your knuckle. If you have that, it's a single gene that you carry that either causes you to have that or not have it. The dimple in your cheek that Michael Douglas here on the right and his father Kirk Douglas have, same thing. A single gene, if you carry it, causes you to have that. A couple of alleles for a single gene cause that. So if you have 2 of those alleles, that's what you have. Otherwise, you don't have that. The ability to roll the tongue, although this one's become a little more controversial lately. Try and roll your tongue like that. Is there anyone who can't do it? Let me see you try. It's always funny to watch people try. It's not that hard, though. Just work at it. No, it doesn't matter. You can work all you want. You're not going to learn how to do it. So somehow, a single gene that you, depending on which alleles you carry for that gene, you either can do it or you can't do it. All right, let's look at Mendel's second law. Mendel's second law, I don't want to spend as much time on because it's relatively easy to understand, but it's hard to explain. So it's something called independent assortment. And in this case, let's say you've got 2 genes we're going to consider. One gene is called the gene for PKU. And you'll read about this in your textbook. Phenylketonuria. I'm not going to spell it out. PKU is a disease that, if you have it, you are unable to convert one amino acid, phenylalanine, into another amino acid called tyrosine. And because you can't convert the one into the other, and then other stuff happens to it, because you can't do that first step, all the phenylalanine that you consume just builds up. And it builds up, and it builds up. And if you get too much of it in your body, it causes brain damage. Yes, it should ring a bell, because if you have drank Diet Coke, does anyone have a can of a Diet Soda in here? I'd love to. On some gum, too. No one has a Diet Coke can. You look, and here's this big warning label. It says, warning phenylketonurex, contains phenylalanine. Why would that be? Because if you happen to carry two of the recessive alleles that code for a bad version of the enzyme that converts it, you don't necessarily have to die. Previously, people did, but now biologists had the idea, well, hey, if you cut all phenylalanine out of your diet, it doesn't accumulate, you don't get the brain damage, you don't die. So now we just test people early on, and if they carry it, they can avoid phenylalanine. Question? So at birth, they test for this. Yeah, and there's some obvious signs early on, but it's a case of nature versus nurture, right? You have the genes, but it depends on what the environment is. If the environment is lots of phenylalanine, then the phenotype is death. If the environment is no phenylalanine, the phenotype is normal life. Question? No, it's not the one where they cut the baby's foot. I have no idea, no. Yeah? Cut the foot. No, it has to do with the urine turning black when it is exposed to air. I'll look into the cutting of the foot. Which reminds me, thank you all for being kind when my son Jack was here on last Thursday. That was like the happiest day of his life. He's been telling everyone that he saw Daddy teach class at UCLA. Okay, so if we do a cross where we've got one individual has no PKU, and not only do they have no PKU, I'm saying that they're dominant for both alleles, no albinism, so they're dominant for both of those. Cross that individual with someone who has PKU, so little p, little m. Those are the genotypes. So how would you do this cross? You'd use a Punnett square. But it's a bigger Punnett square, because the first thing you need to do here block this out. I want you to tell me what kind of gamete each of these individuals produces. Because when you produce gametes, it's through the process of segregation that you end up with one copy of each gene. So draw a little circle that represents the gametes of each of these two parents. It's easy until you try to do it. That's correct. Very good. Yes. So the individual on the left, all of that individual's gametes have a big P, right? But they also have a big M. You can't draw gametes that don't have M. You have to have some allele for the trait. So it would look like this. All the gametes would have one big P, one big M. All these individuals would have one little p, one little m. When they come together, you don't need a Punnett square for this, right? Because every individual is going to inherit from one parent a big P, from the other parent a little p, from one parent the big M, the other that. So what is their genotype of the F1 offspring? They're going to be, they're going to show both dominant alleles, but the genotype is doubly heterozygous. So that's the genotype. The phenotype is no PKU, no albinism. Then draw out which gametes are produced by each F1. This is the hardest thing I'll ask you to do today. Good. So when you're doing the gametes, remember Mendel's law of segregation. One copy of each allele. Almost. No. Remember, every gamete could get the big P or it could get the little p. It could get the big M or it could get the little m. So when you actually write them out, it might have gotten the big P sometime, but sometimes it gets the big M with it. If it got the big P, it might have instead got the little m. But half the time, it's also going to get the little p. But it could get the little p with the big M. Yeah, so could have gotten the little p and the little m. So with respect to these two genes, there are 4 different types. So now if you cross 2 F1 individuals, what are the offspring going to be? It's a big Punnett square, yeah. We call it a dihybrid Punnett square. It's 2 separate traits. But you would just say, all right, we've got this double heterozygote, this double heterozygote. We're going to cross them. I'm not going to do this in class, but it looks like this. Male gametes are on the side. There are 4 of them. Female gametes are on top. There are 4 of them. And there are 16 different combinations that could come through, although you'll see a lot of duplications. Question?
You said before that each offspring receives one chromosome, like one of the chromosomes.
Yes.
If both the albinism and the PKU alleles are on the same chromosome,
Yeah.
limit the amount of
If the big P and the big M, or the P and the M are on the same chromosome, then whatever you have for one, you're going to pass for the other, and it messes with everything. Mendel didn't have this problem because Mendel didn't know there were chromosomes. He thought they were all free floating. So it turns out that in the rare condition where you've got two genes for 2 different traits on the same chromosome, they don't exactly follow this rule. But we're going to ignore it for now. Question in the back. Yeah, mutation, as soon as you get more than a big P and a little p, you have to start using like subscripts. So instead we'd have P sub 1, P sub 2, P sub 3. And yeah, the mutation might be P sub 7, and it produces some wacky other allele. Although already the little p is a mutation, right? It codes for the inability to convert phenylalanine to tyrosine. So you'd fill in all these. I want you to do that at home. Just do it once. And figure out the ratio of offspring phenotypes. And in other words, there's going to be some ratio in terms of whether or not they show PKU or not, whether they show albinism or not. But then they're also together. What percentage have PKU and albinism? Which have PKU and no albinism? Which have no PKU and albinism? And so on. And the reason we do this whole giant thing is to just come up with Mendel's second conclusion, which says that the ratio at which you pass on one gene, or the allele for one gene, doesn't influence what you pass on for another. In other words, you'll see here that if you ignore the M's and you just look at the P, you'll end up with, then it's the equivalent of a heterozygous by heterozygous. It would be 3-to-1. If you just looked at the M's, there would be 3 normal to 1 albinism. In other words, they don't influence what's going on, but then when you layer them on top of each other, you can watch both traits simultaneously, but they're not influencing. So the trait I inherit for eye color doesn't influence whether or not I inherit the ability to break down lactose or not. You'll see when you fill this out and you look at the ratios. The trait that you pass on for one trait has no effect on which allele you pass on for some other trait. It's called independent assortment, meaning that all traits assort out into the population independent of each other. Except for that very rare situation when they're on the same chromosome, then they don't independently assort. The further apart they are in the chromosome, the more they independently assort. And in fact, you can do really cool experiments where you look at the ratio of offspring and how commonly the traits come together, and you can figure out where the genes are. Another question.
But if they're independent, how does that explain blonde hair, blue eyes, or
Yeah, good question. How does that explain some traits that freckles and hair color, the amount of freckles or not, and hair color tend to go along together? And that is because there are some genes that are really close together on chromosomes, and what you pass on for one tends to pass on the other at the same time. So those are rare cases of genes that are close together, but they violate this rule. So I lied. But for the most part, this is true. Here's a question that somebody raised, like in one of the first lecture or second lecture, I think the second lecture, and it's a good question. If an individual shows the dominant phenotype, how do you determine their genotype? That is to say, I have brown eyes, and I told you brown eyes color doesn't get inherited exactly like one gene with two alleles, a big B and a little b, but we can pretend that it does, because it almost does. So if you have someone like me, I have brown eyes. Does that mean that I'm big B, little b? Or am I big B, big B? We don't know. Because the way I color, you could think of it as the little b, like you have a white sheet of paper, and the little b is like a light blue crayon going over it, and then the big B is like a brown crayon going over that. Whether you have one or two big B's, it still looks brown. So how can we figure out what my genotype? What's that? You could look at the parents. So I want to know, I was always curious, am I heterozygous or not? And my mother and father both have brown eyes. So does that mean I inherited a big B from both of them? Not necessarily. Maybe my father is heterozygous and I inherited the little b that didn't show up in his phenotype, and it doesn't show up in mine because from my mother I got a big B. Good question. Yes. So the best way to solve this problem is I have to find someone who has blue eyes, and I have to have a kid with her. And so in the interest of science, I met Julia. She has blue eyes, and we have a kid. Now let's say our kid has brown eyes. Do we now know that my genotype is big B, big B? No, because we know that her genotype is little b, so we know that the kid has a little b, but we know I have at least one big B, right? So I could produce a brown-eyed kid. So what do we do? Have another kid. So you have another kid, she still gives a little b. What do I give? Well, it depends on what my genotype is. If it's big B, I give another big B. If it's big B, little b, half the time I give one, half the time I give the other. And so if we have a brown-eyed kid the second time, what's my genotype? We don't know. So what do? Have another one. If we have a blue-eyed kid, Jack happens to have blue eyes, so first time, we get to stop, right? And now we know my genotype, right? One big B. I have to have passed on a little b. So the tricky thing is, with something like humans, what if we had 10 kids and they all had brown eyes? Can we then conclude that nah? But at some point, I mean, that's like saying, okay, if I flip a coin 10 times and it comes up heads every time, does that prove that it has heads on both sides? If I flip it 1,000,000 times and it comes up heads every time, does it prove that it has heads on both sides? I'd be willing to say, yeah, it's got heads on both sides. At some point, it's so improbable that if it has two different outcomes possible, but you only get one, I have to throw out that hypothesis that there are two outcomes possible. So let me write this down. Here's how you do it. You would say, we see a dominant trait phenotype, so that's me, the big B, and then the line where you don't know it. And so I could be little b, or big B, little b. The cross that helps is with the homozygous recessive. Because think back, if I had started producing children with someone who has brown eyes, that's going to be really hard. That's not going to help me figure out whether or not what my genotype is, right? I mean, it could if we produce a blue-eyed kid, but it's that much harder. When you take the unknown individual and you cross that individual with someone who's homozygous recessive, it's called a test cross, because this individual, called the test crosser, only contributes the recessive allele. So if the person, you don't know what their genotype is, whatever they give, that's going to show up in the phenotype. So if they give a dominant, that's what we'll see. If they give a recessive, that's what we see. So it's really clever, right? That you can figure out what the gamete looks like just by looking at the offspring, because you choose a crosser individual that is going to give two recessives. Question? So given the way I set up the model that eye color is coded for by one gene that has two alleles, could you cross them, two blue-eyed individuals, and get a brown-eyed individual? No. Yeah.
Then if you have one character that's recessive and one that's dominant, you know that the offspring is heterozygous.
Yes. Yeah, exactly. So in the case where we have the brown-eyed kid, we now know that the kid is heterozygous. So in my case, like if my parents, so the first person on here said, look at the parents. If we had looked and my mother had blue eyes and my dad had brown eyes, then we would know, and I wouldn't have had to have any kids. I would know my genotype. Okay, so take-home message number 3 is this. Test cross allows you to determine whether individuals showing the dominant trait are homozygous or heterozygous. It's sort of silly when I give the example of humans, right? Because the thought of producing another kid is really exhausting. Not producing a kid, of raising another kid is exhausting. However, remember, I don't work with humans that often. I usually work with fruit flies. And what's great about fruit flies? They're going to produce 60 offspring all at once. So when I do the test cross, out of those 60, I'm going to expect half of them to show the recessive if the individual is heterozygous. So it's either half or all are going to be is going to tell me what's going on. Question in the back.
What happens when you have two recessives that are different?
Two recessives that are different.
Let's say blonde hair and red hair.
Yeah, there are lots of cases where you have multiple genes for multiple alleles for a trait, and some of those alleles are both recessive. Too complicated to explain in a short period of time. At office hours, I could do it. It's very interesting. I like multiple allele traits. You're all familiar with one that maybe we'll talk about later, which is blood type, because you could have the A allele or the O allele or the B allele. You only have 2 of them, but depending on which 2 you have, some of them, O is recessive, A and B are not recessive to each other. It's interesting and a little bit more complicated. Let's look at the question of sex determination. We all know, I think we all know, that the sex of a baby is determined by the father, right, among humans? Have you heard that before? And do you know why that is? The Y chromosome, the 23rd pair, so we numbered them 1 to 22, and then we have the sex chromosomes. The sex chromosomes are not homologous. In other words, you don't have the same genes on them. The X is on the left side. It's normal size. It's got about 5,000 genes on it. It's big. The Y chromosome on the right side has, it looks just like a kind of a runt of a chromosome. And are there going to be any essential genes on the Y chromosome? Yes or no? Why not? Yeah, you're correct in saying no, because how could it be essential? You're alive. You don't have a Y chromosome. So everything on the Y is unnecessary. I'm not saying that it's not good or valuable. It's unnecessary to have a really nice beach house in Malibu, but it's nice. But nothing on the Y chromosome is essential. Here's how it goes. In humans, you look like this. If you're a girl, you have 2 copies of the X chromosome. If you're a boy, you have 1 copy of the X and 1 copy of the Y. So I've drawn in the color vision gene. So the ability to see colors depends on what you have for this one gene that's on the X chromosome. I've got a little dotted circle on the Y chromosome at the same locus. But you notice that because the Y chromosome is so short, it doesn't exist there. So if you're a male, how many copies of the color vision gene do you have? Just 1. It's on the X chromosome. There are almost no genes on the Y chromosome. I would say maybe 15 genes max. And all of them are going to have something to do with male development, right? They can't have anything to do with something that is necessary to be female, maybe like a barbecue gene or something like that. They're going to be related to that. If they're essential to being a human, they're not going to be there. So you've got this situation. When you're producing offspring, every one of your gametes gets either an X chromosome or an X chromosome if you're a girl. So your eggs all have either an X in them or an X. If you're a boy, half of your sperm have an X in them, half of your sperm have a Y. So then that gets passed on 50-50 because of Mendel's law of segregation blown up not just to genes but the entire chromosome, one or the other. So I want you to ponder this now as a question. Since men have only one X chromosome, are they more or less likely to exhibit a sex-linked? Sex-linked means a trait is on the X chromosome. Are they more or less likely to exhibit a sex-linked recessive trait? I'll get to that in a second. Let's see. Here's what I wanted to look at. So if you look at this in the upper left-hand corner, how many people can see the 29? Wait, I should say how many people can't see the 29? Okay, how many people on the upper right can't see the 45? Couple. Lower left, how many people can't see the 71? Just want to see how many of you are liars. It's not there. I'm making it up. I have to have a control, good scientific method. The lower right, how about the 26? Okay, so everybody can see. Anyway, a standard color blindness test would generally show in a class this size that about anywhere between 4 and 10 males wouldn't be able to see at least one of these, and maybe one female max wouldn't be able to see it. So sex-linked, that color vision trait is a recessive trait. If you have 2 bad copies, you cannot see certain colors well, so you'd be called colorblind. On the other hand, if you are a boy, you only have 1 copy of it. So if you have the recessive allele, you're colorblind. If you have the dominant allele, you're not colorblind. So you get only 1 chance to get the good version. Whereas if you're a female, maybe this one is the recessive colorblind allele, but you get another shot. You can get the good one here so you can see colors. Or if this is the bad one, you get another shot here. So I want you to think about the question of, what if it's a sex-linked dominant trait? All right, running out of time. Let me just go straight to our last take-home message. Sex-linked traits show up in different frequencies in males and females. Sex-linked genes are always on the X chromosome in humans. If something's on the Y chromosome, we'll just call it a Y-linked trait. But they have different patterns of expression. By that I mean different percentages. If I look at blue versus brown eyes in the class, there will be no different in the frequency of that in males versus females. Whereas if I look at something on the X chromosome, I'm going to see males and females exhibited in different frequencies. Okay, so I blasted through a bunch of these. I'll come back next time to show you that in birds and in ants, bees, and wasps and in reptiles, the sex of the offspring is not determined by the male. There's a different way, and I'll talk about that next time. I'll see you on Thursday.